Good morning. Welcome to the session on progress overcoming premature stop codons in cystic fibrosis. We've got 4 really great talks today. So as you know, most of you probably know the premature termination codons, aside from delta F, are one of the more common types of mutations that cause cystic fibrosis or variants that cause cystic fibrosis. And really the problem with premature termination codons, or stop mutations or nonsense mutations as they're called, is twofold, right? So first, of course, the RNA that has premature termination codons in them is unstable. So during the pioneering round of translation, they're recognized as being premature, and they undergo a process called nonsense-mediated decay. It's the cell's way of getting rid of mRNAs that have mistakes in them, like termination codons. And so you're getting rid of all this RNA, and you don't have a lot of RNA substrate there to make a protein. But the small amount of RNA or the protein that is made then obviously is truncated, in most cases non-functional. And so you have 2 problems that you really need to overcome with these. And the way that this is being approached in many cases is, first of all, to inhibit or overcome nonsense-mediated decay, and we'll hear some talks about that, about approaches in stabilizing your RNA, tricking the cell so that it doesn't decay your RNA. And then, of course, once you're making more RNA that can be translated, you're still going to be translating into a premature termination, have that premature termination codon, so still be making more of this truncated protein. And in some cases, those truncated proteins might be amenable to modulator therapies so they can get partial function. But another approach that's being used to make more functional protein from RNA that has termination codons is to promote read-through or to enhance read-through. So that means that the ribosome, when it encounters the termination codon, in some cases, it will incorporate a non-cognate tRNA or amino acid in that position and continue to read through. So you can make a full-length protein, but you're going to have an amino acid in that termination codon that's not the appropriate one. And that's really thought to hold a lot of promise for rescuing variants that have premature termination codons in them. And of course, there's issues around this. What is that amino acid doing in that position? Is that going to be a problem? And so we're going to hear talks that sort of cover all these areas today, and approaches, introducing the problem and approaches for overcoming them. And I'm really excited to hear them. And so without further ado, I'm just going to introduce our first speaker, Emily Bulik-Sullivan, who's going to tell us about pharmacological rescue of CFTR premature termination codons.
So my name's Emily Bulik-Sullivan. I'm a MD PhD student in Scott Randell's lab at UNC Chapel Hill. And I'm talking this morning about one of the projects that I've been working on, trying to find small molecules that can rescue CFTR and PTC variants of CF. I am a grad student. I have no disclosures. So we've already, at this point in the conference, you've probably heard at least 3 people smarter than me describe what PTCs are. I saw a paper recently that increased the percentage of known genetic lesions caused by PTCs up to 20%. So they're pretty important, not just in CF, but also in other diseases. And they have a role as well in cancer, where if they're present in a tumor suppressor gene, it makes intuitive sense that they can allow cancer to develop. And we've already heard a description of NMD. I'm pretty sure I don't need to cover that again. But it's an important target for therapies, potentially. So a lot of people in this room have contributed important work to our understanding of how read-through can happen. So there's 3 different stop codons, which you might remember from undergrad genetics: UGA, UAG, and UAA. And depending on which stop codon is causing the PTC, that can influence the likelihood of read-through. And this seems to vary by drug. So certain drugs are better or worse at promoting read-through at particular stop codons. The nucleotide context surrounding the PTC is also important, as well as the length and characteristics of the 3' untranslated region and the P site amino acid. And so in Scott's lab, Scott and I were talking, and we were saying a lot of this work has been done in overexpression models with reporter assays, sometimes in cells that were not derived from humans. And so we were thinking, it's sort of like the difference between theoretical and experimental physics. We were like, this has all been figured out in a vacuum, but we'd love to see if these rules that we've established apply in human cells in the native CFTR context. And because Scott runs not only a research group, but also a core facility that accepts like donated and explanted human lungs, we discovered that we were pretty well positioned to like work on this project. We actually discovered yesterday that he's been running his core facility for as long as I've been alive. So thank you, Scott, for a lifetime of work. But we realized that between the biobank that he's been collecting since I was born and the CFF's biorepository, we had access to primary human cells, nasal and bronchial, that were homozygous for PTCs and for 9 of the top 60 most common PTC variants in CFTR2. And just, it's kind of incredible that we have these cells in the first place, that there's people who are homozygous for these rare variants, that we have 9 of them. I've actually done some Googling and PubMedding, and I'm not sure for some of these variants if there's anybody who's been found by science who is homozygous for any of the other variants. So it's possible that this might be like a lot of what exists in the world that we have access to. So we were really excited that we had these cells, and we wanted to first make them into cell lines that we could distribute to other people, because it's a rare resource. We don't have tons of the primary cells, and we wanted to make a tool that others could use where you can study CFTR in the human native context, and also perform a small screen to identify drugs that might restore CFTR function in these variants. So then the next step was to figure out which drugs we were going to trial. And this is a great figure that I did not make, but that I wish I had made, showing kind of a survey of all of the drugs that have been discovered to have some read-through activity in any context. Aminoglycosides are their own category because they just have such great read-through potential. And then the rest of these down here are split into non-aminoglycosides. But this was even more than we could really test. The readout that we had decided to use for this project was Tech24 electrophysiology, and our little baby tech machine only has 2 plate capacity, so we can test 48 transwells at a time. And with the number of cells that we were making and the number of drugs on here, we were like, to have adequate sample sizes for each group, we can't test all of these. So we had to do some intelligent prioritization. We decided to prioritize trying to get 1 drug per mechanism of action. We didn't want to like double up and have 2 aminoglycosides when we could just probably test 1. We wanted to prioritize things that had not previously failed in human clinical trials, with the exception of if we wanted to try them in combination with other drugs to see if there was a synergistic effect. And for drugs that had never been in clinical trials before, we wanted to prioritize any that had clinical potential. So if it was known to be way too toxic to put into a human, we didn't really want to waste our time with it. So these were the ones that we initially decided to trial. But even this was a little bit more than we could do for all of these different cell types with the capacity of tech that we have. I also included SMUG1I down here, which is an inhibitor of nonsense mediated decay, which seemed important to test. So we did a lot of preliminary like dose optimization and trialing, and these are the drugs that we decided to reject. Atalurin has failed in clinical trials for PTC variants of CFTR, but we tried it even in combination with other drugs and didn't see a lot of effect, so we got rid of that one. SJ6986 is an ERF3 degrading drug, which is in the same class as CC90009, which I think will be mentioned maybe in other talks. We did a head-to-head comparison, and it was never more effective than CC9, so we didn't include it. And then the aminoglycoside question was a big one, and we spent a lot of time on that. None of these worked, even though the aminoglycosides are toxic, but none of these ones really were any good at read-through alone or in combination with other drugs. ELOX, ELX02 was effective, and about as effective as G418 we saw, but it's kind of expensive and harder to get hold of than G418, so we decided to use G418 as a stand-in, and if we saw effect with G418, we would sort of substitute in ELOX. So these are the suite of final drugs that we decided to test. DMSO is our vehicle control. CC9, a thalidomide derivative, causes ERF3A degradation. Thanks to our friends from Alabama, SRI 413 and 5 made the cut, which is depletes ERF1. G418 was our aminoglycoside. SMUG1I used to inhibit nonsense-mediated decay. And then we added on ETI on top of that to see if, after we promote read-through or inhibit nonsense-mediated decay enough to get a protein, can that protein be modulated? So at this point, we were like, okay, we have our cells. We have made cell lines, BMI1 H-tert cell lines from them. We've picked our drugs. We've optimized our doses. We're ready to just cook and like do this plug and chug experiment. And then all of a sudden, we couldn't grow nasal cells, which was tragic because half of the cell lines that we're using here are nasal. And this is so basic, but it's what I've been asked about the most at UNC internally when I've given these talks, because a lot of people, it turns out, have been struggling to grow cells. So maybe this will help you if you're out there trying to grow struggling cells. We had originally tried growing our cells in pneumocult ALI alone, and as you can see here, these were the 4 nasal codes that grew just the worst. We could not get good electrophysiology from them. And pneumocult, these are the baseline resistances from our tech machine, and you can see that the baseline resistances were 0. So no wonder we weren't getting good electrophysiology. And then, of course, the media condition that worked the best was the most complicated one. There's a clear winner here, where these sort of teal bars worked for all of the codes that we were trying to grow. And this is, we grew them for the first week in 50% F+Y and 50% conditioned media from irradiated 3T3J2 feeder cells, both of which are described in this paper. And then for the second week in 25% F+Y, 25% conditioned media, and 50% pneumocult, and then for the final 2 weeks in pneumocult. And that really coaxed these just struggling cells to grow into healthy enough cultures that we could get electrophysiology data from them. So finally, again, this is the tech traces. You can see we just couldn't get traces from any of these other media conditions. And these all look respectable, if not great. So finally, we had our cells, we made our cell lines, we picked our jugs, and we could just plug and chug and do this whole experiment. And this is sort of our magnum opus, which looks kind of blurry on here. And it's a little bit of a complicated figure, so I'll walk through it. The values in this heat map are the percent of wild-type CFTR force gland response. So we had 3 bronchial wild-type and 3 nasal wild-type donors that we also pretreated with DMSO and then calculated the average force gland response. And that's what we used to figure out like how much of that average force gland response we were able to see with these different drugs. Our cell types are the rows, so separated with bronchial on top and nasal on the bottom. And for each doublet of rows, the top row is the primary parent cells, and the bottom row is the BMI1H turret cell line that we created. And they're paired by their particular PTC genotype, and there are 9 of those. So then just one thing to note is that those 2 genotypes here, those cell lines have been published already, but the remainder of these are novel. And then these are the drugs that we ended up testing and the doses. And you can see like this DMSO column, it's important to note that these values are calculated differently than the remainder of these. These are just the percent force gland response of our DMSO treated PTC variant cells compared to wild type. We've subtracted these values out from here. So this is the DMSO normalized force gland response for these PTC variants. And then now that the layout's kind of more clear, I hope you can sort of start to look at patterns here. So the first 5 columns are our monotherapy conditions. And you can see that there's not a ton of success with any of these drugs alone. CC9 alone for W128-2X and maybe a little bit for G27X did promote a decent bit of read-through, but for the rest of them, not really that much with any of these drugs individually. But then once you get started, so the brighter the color, the better the rescue. And you can see that this group here really seemed to do a lot for a lot of the variants that we were testing. The combination of CC9 and G418 was amazing for like R553X and for G542X. And likewise, we also saw the same effect in G542X with SRI and G418, which was pretty encouraging. This was interesting. We were a little bit surprised that the cell line that we produced a few passages after the primary cells had such incredible rescue compared to the parents. But it's encouraging, too, that generally we see that the primary cells and the cell lines that we made from them are following the same trends of rescue, which is obviously what we wanted to see. I think there's also some failures on this. The E92X cells, we didn't really see any rescue for in any of these conditions, like maybe a little bit here, but it's not much compared to what else we were able to see with the other variants. And likewise, the R75X and R1162X codes that we were using, you'll note they're a single row of each of them rather than a double of rows. We did make BMI1H turret cell lines for those, but those cells were just so weak that the cell lines didn't really survive that well. And we thought it would be kind of disingenuous to include the data because Scott always says you don't get good data from bad cells, and these cells were clearly not very happy. But we still were able to get read-through data for both of the primaries for those variants, which was exciting, and a good bit of rescue for R1162X. So we just thought that this was pretty fun, and we were happy to see it. So we are really encouraged by that we're seeing this synergy between 2 read-through promoting drugs with different mechanisms of action. and that we're seeing it in so many of the PTCs that we tested. It's encouraging to us that this suggests that we might be able to see read-through in other variants that we could test as well. And we're excited to start probing the mechanism by which that happens. We also have these cell lines, 2 of which are published and more of which are forthcoming that we're hoping, we love distributing them to people, so if anybody wants some cell lines, let us know. I just need to finish characterizing them and publish a paper about them. And like I said, we're excited that the trends that we were seeing in the CFTR activity seemed to track between the primary parent cells and the cell lines that we made. And there's a lot, it's nice having this graph finally, because this is kind of just the start. It took like a year to make, but now that we know that there are these drugs that we can use to rescue PTC variants, there's so much more that we can explore. We can start looking at functional consequences of CFTR rescue and ASL dynamics, and looking at cilia beat frequency and trying to figure out exactly why this happens, and characterizing the PTCs so that we know exactly like what the nucleotide context around them is, which stop codon is present, et cetera. And one thing that was mentioned also is we'd love to try in primary human cells to determine which amino acid is replacing the PTC, although that's technically very challenging to do. I'm very grateful to so many people. I could have done none of this by myself, particularly the people who donated their organs and tissues, and the physicians and clinicians who helped to facilitate us getting those cells. Also to Scott's core facility, who are the real champs who actually like process the lungs and the nasal turbinates and give us cells from them, to our collaborators who've provided intellectual input in all of this, and in particular to Scott's research group, and in a special particular to these folks. Jacob pretty much grew all these cells by himself, so I'm very grateful to him. And that's it. I can take questions. Thank you.
So in your triple combination therapy, you had such great rescue, but it seemed like it went down when you added ETI. Do you know why that happened?
Yes, we saw that too, and we were like, what's going on? So one thing that we have talked about a lot is just the additive toxicity of working with multiple drugs. We actually had considered doing a combination therapy in this experiment where we used all of these drugs simultaneously, and we decided not to, because so frequently when we add more than one drug, we just see additive toxicity. And when we add ETI, I mean, these cells were treated with 6 different drugs, these 3, and then ET and I. And so I think it's probably just a toxicity effect, but it's something we saw too.
A couple of questions. The first, I think you touched on this, but the ones that didn't work, do you think that's just the collection, or do you think it has something to do with the particular variants that you're testing?
You mean like with R75X and R116X?
Yeah, or the E92X.
Yes,
Yeah.
it's a good. So I think that's something that we've struggled with, is that when we see rescue, sometimes it's because the cells just aren't growing into good, like well-differentiated cultures, and sometimes it's because they're good cultures, but the drugs just aren't working. E92X, both the primaries and the cell line grow beautifully. They make beautiful cultures, but no matter what we treat them with, we can't see rescue. So I think in that case, it's probably because the drugs that we're using aren't doing the right things for that particular stop codon. Whereas for the R75X and R1162X, I think it's more because the cells weren't healthy enough for them to really be like expressing the proteins that we're interested in.
So regarding that, then, in the beginning, you talked about the things that sort of contribute to being a read-through and the features of the particular variant. Is there, and some of them tend to be sort of look like they're at the N-terminus at the beginning of do you have any comments on why that?
Yeah, I was asked that question at my poster yesterday, and it's a good question. I don't have any particular data that I can reference to answer it, so this is just postulation. I would guess that it's more important which stop codon is present at that particular PTC variant and the nucleotide context around it, rather than its position in the protein. But that is just postulation. Like the fact that we can get decent rescue with G27X, which is so early, that's kind of what I'm thinking. But I really don't know. I have no data to support that.
This is a great presentation that you gave.
Thank you.
And what I was realizing, the first thing, your thanks to Scott Randall. We all have learned a lot from him for all these primary cells. And secondly, I'm very happy that he has started to work on this, and we will learn more. My question, or rather a comment to all of us who are working on these nonsense variants, is that what David Bedwell has shown this repeatedly, that the sequence context is important. And my question comes from one of the very recent findings that got published in Nature Genetics, and you might have read that paper, So what they have done is that they have looked into 5,800 nonsense variants across the genome, and they have tried to look into the sequence context and used eight different nonsense variants. I was talking to David that because I'm presenting my journal club next week, and I wanted to pick his brain on that. So can we, as a community, provide this information out? Because in CFTR is the only protein in which we have a very good functional readout. Can we study all these nonsense variants? It's not possible for one lab to do, but if we collaborate, and then we give a sequence context and read, because if one dude is doing something, another is doing something, and terminus variants we have studied in detail, we can do it in heterologous cell lines, primary cells, bronchial cells, and then give that information out with the functional output. I think that will be really helpful. That is just my comment for this, and this is really good that you are talking about these nucleotides.
Thank you. And I agree. I saw that paper actually just this week, and I was like, I need to rewrite my talk. But yeah, it was interesting because their results kind of contradicted what we did. But they had done what we were talking about, where it was like a reporter assay and an overexpression model for CFTR. And they also didn't try any combinations of drugs, if I'm remembering correctly. They only tried individual drugs and got very different results than what we saw. So I think there's definitely a place for like the synergistic effect of attacking this mechanism from multiple angles that really leads to like good read-through, which they didn't see.
Emily, very nice work. I mean, it was a lot that you had in the slide, lots of experiments. I just a quick question on the ETI where it went down. I mean, did you use ivacaftor also chronic overnight?
Yeah, we did.
So, I mean, you are probably aware of Debbie Codons and other people's works. I think 0.3 micromolar is still at the point where that itself may destabilize the protein enough that you see a decline in function. And actually the elacacaftor and tezacaftor, those you can probably go up a tenfold and you would see more efficacy. So I think it's just the concentration that really sort of sends that down rather than up. So something for the future. But,
I think you're right. If I could redesign this, I think I would have added elacacaftor and tezacaftor for 24 hours and then only ivacaftor acutely. Yeah, but I agree. We were too late and we'd done too many experiments before we thought of that.
So good.
Hi, I mean, it's a very good presentation and it's very beautiful combinations. So I just have one question. Have you ever tried the EF3A and EF1 G418 combinations? Because I didn't see that.
So like, that would be like SRI413 and 5, CC9, and G418. We tried SRI413 and 5 and CC9 together, but we never tried G418 on top of that. These two together are quite toxic. They, for a lot of our weaker cell lines, before we figured out how to grow them, we would just see that the cells had popped off the membrane when we treated with those two drugs together, which is interesting. And we actually initially were working with a much higher dose of SRI413 and 5 and had to reduce it because it was killing our cells so much. But it's a good question. I think we could definitely try that.
Thank you.
Thank you.
So, could I make a comment on that? So, what we do when we do the combinations, that we reduce the concentrations, and what we find is an EC10 of G418 is enough to get a big boost on many of these others. So it's just barely giving them enough to show any read-through, but that synergizes like crazy. So it's like you're priming the ribosome for this stuff to happen.
Yeah, that makes sense. We could use less G418, I think, and still see this effect.
While you're on that topic, I'm always worried about whether any of this is going to translate to patients, right? Mm-hmm. So we know G418 is pretty toxic, and I don't know a lot about that CC 9009, but the one thing we need to also consider as a community is what can we do at low doses like you're suggesting, David, and what can we possibly think if we translate it into doses in humans? It's going to be possible. Otherwise, if we only in cell lines can achieve this, and we get a manuscript that shows we can do this, patients call us and they say, well, look, we saw you got this working. Why aren't you using it in my child? So I think our challenge remains to be, what can we do at the lowest possible dose that might be possible to use in an animal model and show it's safe, and then finally in a human? Because that's the struggle we're still facing after all these years.
Yeah.
And that's why I ask everyone to consider.
The
people are waiting and hoping.
The translational next steps are definitely something that we think about often. And CC90009 has just actually failed in phase 1 clinical trials for acute myeloid leukemia, but it was administered systematically and obviously for a very different disease. So that's actually something I proposed in my fellowship application, was to do intratracheal administration of CC9. Whether or not that will have side effects, I don't know yet because I haven't done that work. But it's a good question for sure. And G418 is definitely too toxic to put into humans, but we really have seen that it works basically equivalently to ELX02, which can be put into humans. So I think another next step would be to try the combination of CC9 and ELX02 and see what you can get. Thank you.
Okay, our next speaker is Jae Suk Yoon from the CFFT lab, and the title of his talk is CFTR PTC read-through can be enhanced by, that's all it says, a transcript-specific ASO approach. Jay.
Yeah, thank you to the organizers for the opportunity to share our recent work on transcript-specific ASO approach for CFTR PTC read-through. Pharmacological modulators that enable ribosomes to read through premature termination codons in the CFTR represents an attractive treatment approach for people with CF affected by PTCs. However, current candidates for PTC read-through have a limited efficacy and/or may cause unintended effects on native stop codons across the transcriptome. This study aims to develop a new approach to safely enhance the efficacy of PTC modulators while minimizing transcriptome-wide off-target effects. During ribosomal translation, various amino acid tRNAs and ERF protein complex, looks similar, compete to bind the cogonate nucleotide sequences at the ribosomal A site. One strategy to induce PTC read-through is disrupt the binding of ERF protein complex to the stop codons during translation termination, giving near cogonate tRNAs a better chance to bind and extend translation. However, as mentioned before, this approach have a potential for the global read-through and native stop codon. Therefore, the transcript-specific approach is important for PTC read-through therapy. It's well established that translation termination can be disrupted by cis-acting elements, such as nucleotide sequences downstream of the stop codon, and structural elements, like RNA stem loop structure. As a role of the structural elements, a couple of studies have demonstrated that RNA antisense oligo structure, duplex structure, or structural elements created by recruiting that Cas13 protein in the downstream region of PTC can induce transcript-specific PTC read-through. The rationale behind the effect of the structural elements is like this. At premature termination stages, stable ERF protein bindings to the stop codon require the recognition of stop codon plus one additional nucleotide downstream. This interaction pulls the nucleotides at position +4 into the A site. However, structural elements downstream of the stop codon could inhibit this process. We aim to explore whether this transcript-specific approach can be applied to CFTR PTC mutations. By the way, a few weeks ago, the Korostelet's lab published a fascinating study using very similar approach to ours, ASO targeting the downstream region of the CFTR G542X mutation, which offers insightful findings. For instance, the effect of their read-through ASOs can be significantly enhanced with their specific nucleotide sequences and nucleotide context, and in combination of aminoglycoside, the G418, or in combination with the suppressed tRNA. Also, they found that this ASO does not affect wild-type protein expression. I highly encourage you to review this paper, of course, after this talk. One key difference between their study and ours is a model system. They used the in vitro translation with a PTC read-through repro mRNAs in a rabbit reticulocyte lysate cell-free system. In our study, we applied this approach in 16HB generated CFTR PTC cell models. Our experiments uncovered the additional and, in some cases, distinct findings. For ASO designs, for ASO designs, we created 22 mononucleotide with the 2' MOE modification at sugaring and PS modification at linkage to enhance nuclease resistance and reduce toxicity and improve cell binding affinity. We screened the ASOs targeting nucleotide position +4 to +21 related to the first base of the stop codon in 2 CFTR PTC mutation, G542X and R1162X. For the delivery of the PTC ASO, we simply utilize free cellular uptake with single dose for 2 days. In the context of G542X CFTR mutation, ASOs targeting the downstream region of G542X, specifically position +9 and +10, enhance the G418-mediated PTC read-through by approximately 3/4 by Western blot. However, PTCs alone didn't show the detectable changes. For reference, these bands shown here are non-specific bands detected by UNC 596 CFTR antibody in 16 HB cells. Since the read-through product represents only 1 to 5% of the wild type, the extremely longer exposure is required to visualize them, which also increase the likelihood of detecting non-specific bands. This PTC ASO approach is also effective for CFTR R1162X mutation. The various ASO targeting the downstream region of R1162X, specifically position +4 to +9, enhance the PTC read-through in the combination with G418 or ELAC02, a synthetic aminoglycoside. Although the data is not shown here, our observation, we observed that approximately 40% of the reduction of ERF3A protein is required to achieve a comparable level of the read-through induced by PTC ASOs. Electrophysiological assay evaluate the enhanced functional activity using 1 or 2 best PTC ASOs in both PTC mutations, G542X and R1162X. However, the increase in the the functional activity is less pronounced compared to the changes observed in the protein expression by Western blot. We believe that the functional activity is affected by specific amino acids incorporated at PTC positions. Interestingly, these PTC ASOs remain effective in a mild reduction in the ERF1 protein. However, it loses the effectiveness under the condition of near complete ERF1 protein depletion. Additionally, this graph illustrates the correlation between the ERF3A protein level and R1162X read-through. Similarly, under the condition of the near complete ERF3A protein depletion, the effectiveness of PTC ASOs is absent. These data and these results highly suggest that the PTC ASO functions by inhibiting the ERF protein complex binding to the ribosome. This RNA-seq analysis shows the extent of the global gene expression changes induced by these treatments for up to 11 days. Compared to the ERF degrader compounds, the most effective PTC ASO for R1162X didn't induce global gene expression changes. Again, these data indicate that highly CFTR-specific PTC ASO can enhance the efficacy of PTC modulators while minimizing the unwanted off-target effects. However, we observed PTC ASO can be associated with different types of the off-target effect. The targeting region of the effective PTCSO we've tested located in the exons. The exon 20, exon 12 for G542X PTCSOs, and exon 22 for R11 Cystic PTCSOs. As a result, these PTCSO can bind to both pre-mRNA and mature mRNA of CFTR. The binding of pre-mRNA of PTCSO in the nucleus can be associated with alternative splicing events. As an example, this Western blot shows both the read-through product and the smaller size bands are ASO dose dependent. Our long-read sequencing analysis indicate that these truncated CFTR bands, truncated CFTR is arise from the transcript variant with exon skipping, exon 12 to 15 skipping. We confirmed using ddPCR these effective R11 Cystic PTCSOs promoted exon 12 to 15 skipping events. For reference, exon 25, 26 junction probe can detect both full-length and skipped transcripts, while exon 11, 16 junction probe specifically identifies skipped transcript. Additionally, the most effective R11 Cystic PTCSO targeting position +4 also induced alternative splicing events, in this case, exon 22 skipping. Presumably, these alternative splicing events reduce the level of full-length mature mRNA, which is the intended target of PTCSO for read-through. As a potential solution, we could redesign these effective PTCSO to minimize their localization in the nucleus where alternative splicing events occur. Alternatively, we could use these types of the ASO, which show only moderate level of the alternative splicing events, and optimize them to enhance their PTC read-through efficiency. We have explored the second option using R11 Cystic PTCSO targeting position +9. The redesigns included adding extra nucleotide at the 5' end to form 5' stem structure, and modifying mRNA ASO base pairing to form stem loop-like structure, pseudonut-like structure, or internal loop structure. Our preliminary results suggest that among those redesigned ones, mRNA ASO duplex containing an internal loop exhibit the most effective. This internal loop ASO can achieve the similar level of the read-through induced by most effective PTCSO, which target the +4 position. Moreover, these internal loop ASO have a limited exon 22 skipping event. As a summary, this study introduced a novel method to enhance the efficacy of current PTC modulators by using highly CFTR specific ASOs. The expected mechanism of action for the PTC ASO is the inhibiting the ERF protein complex binding to the ribosome. PTC ASO can induce alternative splicing events and unintended off-target effect. However, we observed further optimization of PTC ASO design could improve the efficacy while minimizing the induction of alternative splicing events. I'd like to thank every member in CFF lab, and special thanks to the CF community for support this study. And thank you, and I'll take your question and comment.
So I can start. It was interesting, the one with the internal loop worked the best, right?
Yeah.
Why? You got a idea how it might be working?
Honest answer is that I do not know. But based on the study, the screening of the structural elements in the downstream area of stop codon, one of the factors affecting the read-through is the bulge structure in the stem loop structure. So we just try to mimic of the structure using the mismatch of the base pairing. But in terms of the mechanism of action behind that, I don't know.
So it smells like it might be the stability of the duplex. And so if you put a bulge in the middle, maybe a helicase can push through it, or the ribosome. Maybe the ribosome is having trouble getting through your duplex.
I know you said your low molecular weight band was due to exon skipping, but do you think you're also preventing the formation of the SURF complex if you don't have ERF1 accommodation? Are you seeing like truncated protein too that would be the length of like G542X truncated? Are you stabilizing the mRNA?
I'm sorry, I couldn't get your answer.
So
I couldn't question.
without the release factor, you're inhibiting the release factor bonding. So do you think you're inhibiting NMD by inhibiting the release factor bonding because you can't form the SURF complex?
Yes, it's also possible. I mean, in terms of the exon 12 to 15 skipping, it's naturally occur. So it's very possible that if under the condition of NMD inhibiting, it's more chance to get the exon 12 to 15 skipping events.
Did you see a large like mRNA level stabilization?
I mean, it's not huge, honestly.
It's really nice, Jay.
Sure.
So in your studies, were you surprised that you never reduced the wild type levels of protein? And then did you ever design ASOs in which you stalled translation in just a wild type transcript? I think that I always thought that this approach promising for nonsense mutations may affect actual just translation rates. So when you compare, so did you ever make an ASO that reduced wild type expression of CFTR?
All right. So when we used the wild type 16HBE, we could see the reduced level of the full length of a wild type. It's because of the splicing events.
And did
Wild
those
type
differ
splicing
between
events.
your knots, your internals? Could you really make was there any designs that was just flawed because the wild type wouldn't express using that approach?
So I mean, before that, the bottom line is based on the in vitro study.
Yeah.
This static blocking ASO does not affect translation elongation rate.
Yeah, I guess that's shocking to me, but yeah, that's what it is.
Because it's the kinetic of the translation during the elongation and termination is different. The translation termination, in the kinetic of the translation termination is much slower. So it's a better chance to get the ASO can kick in, but the kinetic of the elongation is relatively fast. So, and displace of the, annealing efficiency of the ribosome is quite high. So even though the static blocking is blocked, typically the translation elongation is not that affected.
That was really beautiful. I love the specificity and the lack of effects of the drugs that degrade ERF1 or EF3. It's really profound. But the question with oligos will be delivery.
Mm-hmm.
So how do you anticipate this could work in vivo? For example, would systemic delivery, which would access the basolateral side of the cells, which is more active in endocytosis, would you be able to reach the concentrations that you need to have the effect?
Yeah, that's a great question, and honestly, most hard question to answer. Yes, that is the truth. We need to that's the biggest part we need to overcome and to test it, and particularly in terms of the concentration in the target cell. Reach the concentration, proper concentration in the target cells is the biggest. Yeah. Honest answer is I do not know yet, but we're gonna try to push it. Yeah.
Gary.
Yeah, perhaps you mentioned it, but what do you think is the mechanism of exon skipping induced by these ASOs?
Michelle is the expert for this area, so I guess that there is.
You're as puzzled as I am, so I can't yet think of how that. I can envision a few things. I know nonsense variants have been reported by us many years ago to actually occasionally induce exon skipping, but that hasn't been replicated much. Maybe R553X has been shown by Ann Harris to be skipped every so often, that exon containing that one. So I'm wondering if there's a connection somehow, and we could learn why exons are excluded occasionally. But it's very interesting what you've shown, that these ASOs are causing that effect in addition to the one that you're looking for, which is read-through. So, but I can't,
So
somebody else has got an idea. I'm trying to think how
one of
exactly
the
that
data-
exon skipping is occurring. How's it interfering with the splicing mechanism?
One of the data we have is the G542X mutation transcript has the higher exon skipping compared to the wild type. So I think definitely the one single nucleotide change affect the binding of the factor related to the exon skipping. I'm sure it's so probably also the PTCs binding to the region near there also affect the binding of the factor related to the splicing events. That's probably the best answer I can have.
Okay, so if there are no other questions, let's thank Jay.
Thank you.
Okay, the next talk is by Anna Borelli, and her title is Rescue of CFTR Nonsense Mutations is Enhanced Under Inflammatory Stimuli.
Good morning, everybody. I am Anna Borelli, a fourth-year PhD student in Luis Galletta's lab in Italy, and I'm very happy to share some of the results of our work. And so for this, I want to thank the organizers for selecting my abstract for the oral presentation. I have nothing to disclose. And to date, as all of you know, CFTR pharmacotherapy has been the leading strategy to treat the majority of people with cystic fibrosis. In particular, the triple drug combination named the Trikafta or CAF trio in Europe is able to correct the CFTR basic defects in patients with at least one copy of the F508 deletion mutation, that is the most frequent mutation. It combines three different drugs that act on different sites on the protein and improve and correct the folding stability and gating defect caused by this mutation. However, there is a substantial percentage of people, ranging from 10 to 30% according to the geographic area, that cannot benefit of this highly effective modulator therapy. In particular, people carrying class 1 mutations are one of these. Class 1 mutations includes nonsense mutation that introduce premature termination codons, also known as PTCs, in the coding sequence of the protein, thus leading to an abnormal truncation of the CFTR channel. PTCs also cause the degradation of the mutated CFTR mRNA by the nonsense-mediated mRNA mechanism. Together with the W1282X, the G542X is the most common mutation of this class. A possible pharmacological strategy to overcome premature truncation is the use of read-through agents that act on the stalled ribosome, decreasing its proofreading ability and increasing the possibility to insert a near-cognate tRNA in the place of the premature termination codon, thus allowing the continuation of the protein synthesis. Aminoglycosides like G41A and gentamicin were the first proof of principle read-through agent, but their high toxicity impedes the real application in vivo. So safer aminoglycosides like ELOX2 were synthesized, and this compound is presently under clinical investigation for patients for people with cystic fibrosis. Recently, a small molecule known as ERF3 degraders has been shown to promote rescue of CFTR-bearing premature termination codons. In particular, this compound, the CC90, is an ERF3 degrader that acts as molecular glue, enhancing the affinity between proteins. In particular, CC90 is a cerebron E3 ligase modulator that induces the proteasomal degradation of the ERF3 factors that has a fundamental role in the termination of translation. Recently, it has also published that application of inflammatory stimuli in vitro on bronchial epithelial cell can also enhance the rescue of delta F508 CFTR rescued by CFTR modulators. So we asked if this inflammatory condition, this inflammatory stimuli, can also influence the pharmacological correctors of CFTR that bear premature termination codons. To answer this question, we generated fully differentiated bronchial epithelial cells from patients carrying G542X mutation in homozygosis and studied them with short-circuit current recording technique, where we can maximally stimulate the CFTR chloride secretion by an analog of cyclic AMP, then further potentiate it with the CFTR potentiator, and then inhibit fully block the current with a specific CFTR inhibitor that is the 172. Then we can measure the extent of CFTR correction by measuring the drop of current elicited by the inhibitor. So, as you can see from the representative traces and the dot plot, the application of the read-through agent plus a class 1 corrector did not elicit a statistical significant correction of the CFTR function. However, the situation changes when we apply the CC19, that is the ERF3A degrader, in which we can reach a threefold correction of a CFTR chloride channel comparing to untreated cells. So we decided to test these two combinations of drug with two different inflammatory stimuli. The first is the IL-4, that is a Th2 cytokine that induces goblet cell hypersecretion with the abnormal mucus production and increased chloride current depending to the CFTR and TMM16 channel. Interestingly, when we applied IL-4 for 72 hours, we saw an increase of 8-fold of the rescue of the CFTR function comparing to the untreated cells when we treat the cells with the read-through agent and class 1 corrector. This rescue further increased to 15-fold when we also applied the ERF3 degraders, as you can appreciate from the large increase of current in response to the cyclic AMP and the terrific drop of current elicited by the inhibitor 172. Besides, we also test the combination of IL-17, that is a Th17 cytokine, plus TNF-alpha, because these two cytokines are characteristic of people with cystic fibrosis. And surprisingly, we found that, yes, the application is still effective in rescuing CFTR, but only when we have the combination of the ERF3 degrader, the read-through agent, and the class 1 corrector. We also confirmed this data on a different patient carrying the G542X with another mutation that is not sensitive to pharmacological treatment. And we observe a similar extent of CFTR rescue. We also noticed a decrease in the function of the ENaC and TMM16 channels that is probably due to the application of the CC19, as also previously reported. We also confirm, moreover, the same result on nasal cells with a third genotype. So, given the results at the functional level, we also asked what happens at the protein level. We use capillary-based protein separation and immunodetection that provides a good sensitivity. On the left part of the slide, you can see the chemiluminescent signal profile represented as a virtual Western-blot image. And we can clearly appreciate the appearance of a strong signal corresponding to a fully mature form of CFTR in the samples where the triple drug combination is combined with the cytokine treatment. The signal is quite dramatic for the IL-4, but if you look at the quantification of the area under the curve for the signal, also the application of IL-17 plus TNF-alpha reaches the statistical significance. We also looked for the expression of the ERF3A protein, that is the target of CC19, and as expected, the signal completely disappears upon treatment with the CC19 compound. Interestingly, also cytokine modestly but significantly lowered the amount of the ERF3A protein. So we analyze the effect on stability of mRNA by real-time PCR. And as you can see from the graph, the triple drug combination did not alter by itself the amount of the transcript, but it increased upon 4-fold and 6-fold after the application of IL-4 and IL-17 plus TNF-alpha. So we analyzed the function, the protein, and the mRNA level, but what happens at the airway surface liquid? So with the fluorescence recovery after photobleaching experiment, we can measure viscosity, we can assess viscosity of the epithelia by treating the apical side of the cells with a fluorescent Fitch dextran. Then we can with an spinning disk microscope, we can photobleach selected circular area and then measure the recovery of the fluorescence in the photobleached area due to the diffusion of the surrounding fluorescent dextran as a measurement of viscosity. As you can see from the image on the bottom, on the right is depicted an high viscous epithelium that is characterized by a very slow recovery of the fluorescence. So with this technique, we demonstrated that application of IL-17 and TNF-alpha on bronchial epithelial cells induce an hyperviscous state that can be switched to a more fluidic state upon the activation of the CFTR with beta-adrenergic stimulus. This is also obtained when we treat the delta F508 mutation with the CFTR modulators. So we asked if also the pharmacological correction of the G542X mutation is enough to obtain this switching in viscous state induced by the combination of these two cytokines. As expected, with the triple drug combination, epithelia treated with the combination of IL-17 and TNF-alpha showed a significant reduced viscosity, as you can see from the upper part of the graph, in which are depicted the fluorescence recovery after photobleaching during time. Without the triple drug combination, there is a very slow recovery of fluorescence that is recovered when we apply the triple drug combination. That I remember it's composed by an ERF3 degrader, a readthrough agent, and a class 1 corrector. Surprisingly, when we treat epithelia with IL-4, we don't see any difference in viscosity in respect to untreated cells, even with or without the triple drug combination. And this is quite surprising since IL-4 induce a very abnormal mucus hyperproduction. So, in conclusion, we demonstrated that inflammatory condition can also improve the rescue of CFTR-bearing premature termination codons. And this is quite interesting because this pharmacological treatment could be more beneficial than expected in vivo. This data also stress out the necessity to investigate more deeply how these cytokines can modulate processes like readthrough and NMD, and this could reveal novel targets that can be modulated with pharmacological agents or antisense oligonucleotides. So I want to thank our funding agency, the Cystic Fibrosis Foundation and Italia Foundation for Cystic Fibrosis, my mentor Luis Galietta that given me the opportunity to work on these interesting topics, and my colleagues that now are more colleagues, they are friends, in particular Arianna Venturini and Martina De Santis, that share with me everyday life and experiment, our external collaborator, Dr. Fabiana Ciciriello, for giving us the cells, and all of you for your attention. I'm happy to take questions and suggestions.
I can start. So that was very impressive.
Thank you.
A guy named Roberge in British Columbia published a few years ago aminoglycoside enhancers. They didn't have much effect by themselves, but they really enhanced the aminoglycoside mediated read-through. And he showed that those were cytokine dependent. Mm-hmm. So I wonder if you've got something like what he had, but it's working with CC90009. And what he suggested they were doing was increasing ribosome biosynthesis as well as translation rates. So that might be something you should think about.
They are probably perturbing something related to ribosome.
Right.
behaviors. I mean,
Yeah. So that's interesting.
yes.
Well, that was very nice work. And, we all think that inflammation in the CF lung is a bad thing, but it's kind of amazing that it can enhance the activity of these read-through compounds. Do you think that a mechanism is that there are more of the secretory cell types in those cultures exposed to the cytokines that can effectively, process and create functional CFTR? It would be kind of interesting to do a single cell analysis of the cultures after the treatment and see if it really is the appearance of those specific CFTR-rich secretory cells that are
Yes.
have an effect.
A single cell sequencing is the next step we are planning to do, obviously. And I really don't know if we can alter the cellular composition in only 72 hours of treatment. My very personal feeling is that we are doing something also on mRNA stability. We inspect the previous sequencing data and that were made on non-CF cells treated with cytokines, and we find interesting candidates like poly-A binding protein C1, PABC1, that also has a role in nonsense mediated decay. So maybe we can start, before to having this sequencing data, we can start to evaluate the most important key factors in participating in NMD and ribosome dynamics.
Yeah, and I'm sure someone's going to ask, did the cytokine treatment induce ionocytes in the cultures?
Yeah, we have to check.
Thanks. That was really interesting. There's a lot to unpack. A lot of clues, which I love, getting some mysteries there. But
yes.
So I was curious, I guess the one, since I like to think about splicing a lot, the 1717-1, right? That's an interesting,
yes.
You said that one was recovered, right? And so do you have ideas about the mechanism? Because relating to Scott, I would love to see, look at RNA splicing and the transcripts that occur after this, right? Because that would suggest that maybe there's a cryptic splicing that's occurring and making a stable, like you said, stabilizing the RNA perhaps and making a protein that is being recovered. In some...
Personally, we don't have check yet because we didn't have time. We only searched for other genotypes with other mutation and observe if the behavior was the same. And yes, we have to check. Yeah.
Yeah, because your last point on your conclusions is revealing new targets. It's absolutely correct, right?
Yes.
So if we can look at these and see what's happening, maybe it'll help us to...
I think it's the most exciting part of the research.
Yeah, absolutely.
Hello, great talk.
Thank you.
I have to admit I know very little about cytokines after seeing your poster yesterday.
Yes.
You could comment on the fact, looks like there were works where people claim that treatment with cytokines without G542X or any other CF context in general changes expression of mucins and viscosity. So in your work, yes, you directly show the function, no question about that. But when you talk about viscosity, how much of that is CFTR per se versus ENAC? Did you do wild type cells to see change in viscosity? Can those be deconvoluted?
Yeah. I think the IL-4 behavior is interesting in this case because we know that IL-4 induces very broad changes in the epithelium because it increases lots of the mucus and also decreases the ciliated cell composition of the epithelia. But strangely, we don't see any difference when we apply the drugs to the cytokine treatment. So maybe they are not interdependent.
In wild type, without G542X context.
In wild type, okay. No, we have to check. Yes.
Okay, I think I had the same question, which was just what happens if you try this in a wild type? Is it specific to a transcript that's undergoing NMD, or, if you don't have a transcript, do you...
Yeah, I think it's something that is related to the nonsense mutation because in wild type, you don't see such enormous differences between in viscosity, I think. But I have to check. I have to check.
Okay, but have you looked at like wild type transcript abundance or anything like that after?
Yes, I think if I'm not wrong, also in literature we know that cytokine can also increase mRNA levels by CFTR itself.
Okay, great.
Have a time.
Did you say that after cytokine treatment, the G542X mRNA went up fourfold or something? Was that
Yeah,
what it was?
with IL-4 fourfold and IL-17 and TNF-alpha sixfold.
Sixfold, yeah. So the question is that PTC specific or not?
It seems that is
So it is.
PTC specific.
So
yes.
that looks like NMD then.
Yeah. Mm-hmm.
Can I ask a question?
Sure.
Oh, thank you. Again, bringing the same point on wild type. The data that you showed on using these read-throughs, and you talked about the amyloid response, and that question can go back to the previous talks as well that Scott's student showed. When the amyloid response goes that drastically low, number one, do you consider this as a good news or a bad news? And secondly, I haven't seen yet that when you are using these compounds, if you tested this in a wild type, or even to make it a more convincing point, can we do it in a carrier individual who has a wild type allele and a nonsense, and you just use as a control to demonstrate that at least with these variations in the ENAC, you did not alter the wild type currents?
Yes, it's very interesting point. I think the effect on ENAC are not specific effect of the drug. Probably they are off-target effect, but they can be, I think, exploited in some functional ways because obviously you are depleting cells for sodium content. So maybe they can have good beneficial effect. We didn't have tried yet to the wild type, but it's one of the next things we surely do.
Mm-hmm. Okay, so if there are no more questions, let's thank Anna. All right, the next speaker is Carrie Thrasher, and her talk is entitled Determining the Functional Consequences of Translational Read-Through at Common CFTR Nonsense Mutations.
Okay, I have no relations to disclose, seeking disclosures, I guess, at this point. Okay, as multiple people have mentioned in this session, a nonsense mutation is a point mutation in the DNA that results in an in-frame premature termination codon, or PTC, in the mRNA. And so far, there have been around 150 different PTC alleles that have been identified in people with CF, according to the CFTR2 database. And this really makes up around 10% of all disease-causing alleles. However, this is the 10% that is unresponsive to currently available modulator therapies. So the reason why it's not subject to modulation is because nonsense mutations really disrupt the flow of genetic information through two distinct yet related processes, including nonsense-mediated mRNA decay, which I think we'll hear about in the next talk. Also, we have premature termination of translation of any RNAs that are able to escape NMD. So that's really the focus of this talk, is how we're going to inhibit the premature termination. So normally when the ribosome encounters the PTC, shown in red here, you have accommodation of the eukaryotic release factors, ERF1, and its associated GTPase, ERF3. And upon accommodation, you get release of the truncated protein that is usually non-functional and rapidly turned over. So to overcome nonsense mutations, our lab really focuses on exploiting the mechanism of translational read-through. This is an endogenous mechanism that really only occurs at very low levels in the cells, where instead of ERF1 and ERF3 accommodation, you have accommodation of near-cognate tRNAs. And by near-cognate, that just means you have anticodon-codon interactions at 2 of the 3 nucleotide positions, so you will get a mismatch at one of the positions. Shown here in this little diagram box, there's a mismatch at the 3rd, or what's traditionally called the wobble position. But upon that accommodation, you have the associated amino acid to that near-cognate tRNA in the elongating polypeptide, and then translation can continue in frame for the entirety of the coding region. You can induce this read-through mechanism to a higher frequency using small molecules, several of which have been discussed in the previous talks, including adelurin, the release factor degraders, some tRNA modifiers, and aminoglycoside antibiotics such as G418, which is what I'll be talking about today. However, there are some consequences to read through to think about. So if you get accommodation of the near-cognate tRNA, you can have a mismatch at really any of the 3 nucleotide positions, which leaves you a pool of near-cognate amino acids that can be incorporated into the elongating CFTR protein that might not be wild type. So at any given stop codon, shown here in the stop sign or the 3 different stop codons, you have 6 to 7 different amino acid possibilities that can be inserted. So that raises the question, are we creating CFTR variant proteins? And do those variant proteins have reduced function compared to CFTR wild type? So to look at this, here's the workflow of the project. I can induce read-through using G418, then purify the read-through protein, perform tandem mass spec analysis to identify what amino acid was actually inserted upon read-through, and then determine if that variant is actually going to be functional, and if it does have a protein dysfunction, can that dysfunction be modulated? So here are the different PTC alleles used in this study and ones that we have previously published. These represent all 3 stop codons and are some of the most common CF PTCs seen in patients. And they also span the length of CFTR. However, as was alluded to in the first talk, CFTR is a very low expressed gene. We also have to contend with NMD. So purifying the full length read-through product is a very technically challenging problem. So we developed this reporter system where we have a turbo GFP upstream cassette, in which downstream we have a CFTR cassette, which includes the CFTR PTC mRNA context. And then downstream, we have an HA tag and a hexahistidine sequence that we can use for purification. So only if we read through that CFTR PTC cassette would we make our purification tag, and that would be the protein that we would perform mass spec on. So the mass spec data shown here are the new alleles that we've discovered, and we have all 3 stop codons, UGA, UAG, and UAA, and I'm just going to hit some high points here. So the R553X is similar to what we previously published with the G542X. This seems to be the common motif at UGA stop codons. However, R1162, we had almost no ARGs insertion, which would be bad news, as that would give us a wild type protein. However, better news is at the Q493X, we get 99% glutamine, which would give us a wild type protein. So there are some alleles where we not only get some fraction of wild type CFTR being produced, but a lot of times, sometimes we actually get the majority of the read-through product would be wild type CFTR. E60X, the most predominant amino acid being inserted is serine, which is actually a mismatch in the second position of the codon-anticodon interaction, which is thought to be a very rare event. But the Y122X and Y1092X, the UAAs, are similar to what has been previously published at UAAs. So now that we know all the protein variants that's being produced upon read-through, we can make those CFTR variants and perform functional analysis. So these are all the variants that I identified in the mass spec. The ones in bold are actually wild type CFTR, so we don't need to analyze those. And to perform these function assays, we did TEC assay, or trans-epithelial chloride conductance, which is a CF functional assay where you can add forskolin, a cyclic AMP agonist, to activate CFTR channel activity. And we can also utilize the CFTR-specific inhibitor INH172 to confirm its channel CFTR specificity. Also, as you know, we've now made a full-length protein product that is subject to modulation, so we can also treat these CFTR variants with the currently available ETI therapies. So ET was treated chronically, and ivacaftor was used acutely in the middle of the TEC experiment as designated by the AVA. So this is a lot of data looking at all these variants. All of it was done minus and plus ET, so there's quite a few conditions on the screen here, so I'm just going to cover some high points again. The bad news is all of the variants in this study had reduced activity compared to wild type. That's not always the case. We have identified variants produced upon read-through that have no functional defect and some that are actually hyperactive variants. However, 8 of the 11 variants that we in this study were able to reach wild type level of activity when treated with ETI. One such case is shown in green, the Y122K, where in purple is without ET, and with the navy blue, you can see the navy blue line on on top, that's with the ET treatment, so you get a robust ETI effect. However, some responsive were not responsive to ETI at all. One such case would be E60X in purple on this graph. So it's basically at the axis, and the navy blue line directly above that is in the ETI therapy. So not all CFTR variants are going to be modulatable that are produced upon read-through. Interestingly, some variants were responsive to ET but unresponsive to ivacaftor. One such case is the R1162C highlighted in orange, where in the yellow line is the no ET, so just the base effect of the variant, and the plus ET is the red line. My mouse disappeared. I don't know where it is. Okay, red line right here, where you see an ET effect comparing yellow to red, but no effect before and after ivacaftor addition. So we can then take the exact same cells that were used in the tech assay and perform Western blotting on them to look at if abundance of CFTR is the reason for any functional defects that we see. So is the CFTR variant inherently unstable or not able to be processed to the cell surface, and that is what we observed. So ET did not rescue CFTR expression of some variants. One such case is the E60Y shown in red, where that variant just seems to be inherently unstable, and ET did not help that. Some variants, however, were ET robustly corrected the expression, such as the Y1092K variant, where you can see a large change in the minus and plus ET condition. However, the better news is some variants were able to be processed well without ET, so they had no processing defects, including the Y1092Q, where in the minus ET condition, abundance was equal to wild-type CFTR. So next we wanted to look at why we were observing these differences in amino acid incorporation. So these were all performed in the same cell line. So tRNA abundance wasn't really a factor in the differences in amino acid incorporation that we were seeing here. The only thing different about these reporters were the CFTR context itself. So obviously that was having an effect on what amino acid was selected upon read-through. So to look at this, we took two of the alleles we previously published, G542X and W1282X, which has striking differences in what amino acid was incorporated upon read-through. So at G542X, you get cis, trip, and ARG, where W1282X, you do get cis and a little bit of trip, but mostly you get leucine insertion. So we wanted to know why leucine was selected at one and not the other, when the only thing different was the native mRNA context surrounding the common UGA stop codon. So to do this, we had two new reporters that we created with the upstream G542X, but we switched downstream of the stop codon, the W1282X, and vice versa with the W1282X context upstream of the stop codon, but we stuck the G542X downstream. When we did the mass spec analysis in the GXW, it mimicked the G542X, indicating the importance of the upstream CFTR context in selecting what amino acid was inserted. So just having the W1282X downstream of the stop codon did not induce incorporation of leucine. Also, the WXG, however, we did not get leucine incorporation by just having the W1282X upstream context. So we did also need the downstream W1282X context to mediate leucine incorporation. So that leads me to my conclusions. Where wild-type protein is being produced upon read-through of some CFTR PTC contexts, so 4 out of the 6 in this study, we did at least get some wild-type protein being produced. However, CFTR variants are also being produced. The good news is out of the 11 CFTR variants that we analyzed in the study, 8 were able to reach wild-type levels of function after ETI treatment. However, they did have a base defect. And in generally, protein abundance, which is a measure of stability and trafficking, correlated with the CFTR function. And both upstream and downstream CFTR mRNA context affected what amino acid was actually incorporated upon read-through. And with that, I would like to acknowledge the members of my lab, Matt Renfro at UAB, who lets me use his mass spec anytime I need it, the CF Research Center at UAB, whose core facilities, including Carl and Jay, have been really instrumental in getting this project together, and our funding sources. And I'll be happy to take any questions.
Great, that was really interesting. Two things, obviously two things that were there for me was, okay, so determining, right, without having to do all the experimentation, whether or not you're going to make a protein with what you got there, right? Have you been working on trying to do that prediction kind of through maybe more like AI, like looking at the structure that's made, how you might disrupt interactions with the modulators and things like that, so we can sort of do predictions rather than all the experimentation?
Yeah, I mean, there's some trends that we've seen. So I don't think the data that we have enough right now is enough to, do like a machine learning model with this limited data set that we have. But we've noticed sort of the domain or the location of the protein is very important in determining whether or not it's going to be amenable to have a functional variant. So as someone mentioned earlier, like the N-terminal one, it seemed like the E60X variant that we had, those were the ones that were unmodulatable, would not create a CFTR variant that was functional. So we think the location of where it is, it's going to be important. That was also indicated in one that we previously published, the G550 W variant, which was created upon wild type, was hyperactive, and that's just because of where it is in the LSGGQ motif. So I think it's going to be the location of the variant as to whether or not you're going to produce a functional protein. So that's function, but as far as identification, we just don't have really enough data to make any robust observations in trying to predict what amino acid's going to go insert.
Yeah, or what the function of the protein is after, with whatever and when you get. And then my other question, I can let somebody else go, but really quickly. So I guess we've been learning a lot about sort of structure, maybe around stop codons that might be influencing things like, incorporation and read through and things like this. Have you looked at structures that might be incorporated in, I think
like RNA secondary structure.
Yeah.
We've tried, that was just like running it through like a RNA structure prediction. And like I said, just with the 6 alleles in here, there wasn't really anything that stood out that we could make because a lot of times we have commonalities, but then the RNA structure would look different. And so we didn't really be able to correlate one versus the other. Because there were quite a bit of commonalities between the alleles. There were differences, but there were commonalities, but the structure wouldn't necessarily look the same.
Yeah, cool. Yeah, we've identified a structure through shape analysis because we work a lot on W1282X, and there does seem, it was cool to see that you saw that downstream important region because we see a unique structure that's created by the W1282X.
Yeah. And I think a lot of people are looking at also velocity of the ribosome, so like the sequence structure, so that could also play a role.
David.
So that was very nice. So it was a bit fast for me, but so the R1162C you said was unresponsive to ivacaftor. Is that correct?
Yes.
So that's going to be away from the ivacaftor binding site. So how do you interpret that result?
Yeah, so we're looking at that, especially because that's not the only variant that we're kind of confused about, like the unresponsiveness to ivacaftor, but we're doing some modeling, some molecular modeling to try to see, using PAML. I'm not a structure person, but we can throw that variant into PAML and see if what it looks like. But that's kind of challenging. But yeah, I have no idea why that one's not. I have the same question about the E60X versus Y122X, where it's always sort of hypothesized that the Y122X can reinitiate downstream is why we get so much function with the Y122X, but I do not see that with the E60X, which theoretically would also reinitiate. So I don't know. There's still some challenges.
Thank you very much.
Could it be possible that one is more responsive to forskolin, so there's less left at the end for the...
Yeah, we are using... Yeah.
So coming to the same point of when you are creating this, or we are creating this as a journal statement that there are N-terminal variants like E60X, it's non-modulatable. I think this thing we are saying based on the drugs that we have currently available. I strongly believe that for treatment of nonsense variants with the read-through, what we need and what CF has told is that modulators are very important. So if we are saying that ETI is the standard for suggesting that what are modulator responsive is not correct. Next generation of the modulators that are coming up, you call it VTI or next generation I-20, 50, 70. I think all these missense variants can be modulatable. What we need to ask is to keep on looking for these screenings to come up not only for F508del, if somebody has the database that every variant in CFTR, if you create a missense, which of the drugs are going to work? I think that will be the precision medicine, and the cornerstone for the read-through drug would be an addition of modulators. So we need more people like Vertex to give us these compounds.
Hi, I was wondering if you had tried this out in a different cell line that had different tRNA abundances, and if you saw a difference, or if the difference is just the sequence context.
Yeah, so the differences here are obviously not tRNA abundance because it was the same, but we are doing it in 16 HBEs right now. But we're also doing long read sequencing to actually get the tRNA abundance in the 2 cell lines where we can correlate tRNA abundance with amino acid incorporation.
Great, and then I also had a question if you had tried out any other of the read-through agents to see if that had any influence.
So I've tried some with the same mechanism, so ELX02 compared to G418, and saw the same thing. And I also tried one with a different mechanism, so the ERF1 degraders, and actually still saw the same thing.
Okay, I mean, that's not actually that surprising. It's probably codon, anti-codon binding, but Okay, great.
So far, no differences, but you never know,
All right, if there are no more questions, let's thank Carrie. Okay, for our last talk today, we'll have Zhongyu Li. His talk is titled Global Suppression of Nonsense-Mediated mRNA Decay Exacerbates the Inflammatory Response.
Good morning, everyone. And first, I would like to thank David and Michelle for giving me this opportunity to share my work. I'm work with at Jagannathan Lab at University of Colorado Anschutz campus. I have nothing to disclose. I hope I have a long list here. First, please allow me to use this oversimplified diagram to give you a brief introduction. What is a MND? Let's say we have a transcript, which is CFTR mRNA here, and because it contains nonsense mutation, now we have a PTC here. When ribosome walk through this transcript trying to make CFTR protein, when ribosome sees PTC, it stops here and starts a lot of MND factors being recruited, including UPF1 and SMUG1. SMUG1 is a kinase which phosphorylate UPF1, and the phosphorylation of UPF1 will further stabilize this huge complex and recruit more and more factors. At the end, including these factors SMUG6, which is endonuclease, and RNase, which will degrade this transcripts. And as you can imagine from this whole sessions that inhibit MND is a promising therapeutic strategy to increase CFTR protein abundance. However, it might be, we need to be cautious. And also because SMUG1, it's a kinase, so now we have this small molecule that is highly potent and highly selective to inhibit SMUG1 activity, which I heavily depends on to collect all my data that I'm going to show you today. And the reason that I said maybe inhibit MND to help CF patient, it's not 100% a good idea. It's because MND not only surveyed and degrade aberrant transcripts with PTC, it also regulates some sort of normal transcripts. Those normal transcripts, they don't have PTC mutation, but just because they have upstream open reading frame, 3' UTR intron, or they even have long 3' UTR, they can also trigger MND to target those normal transcripts. And by estimation, up to 20 to 30% of normal transcripts are regulated by MND pathway. So inhibit MND globally might cause some negative effects that we don't want. And in fact, there are several public available data sets out there. And the main purpose for those experiments that researchers try to identify so-called MND substrates. For example, in the Venn diagram that I'm showing you here from these 3 independent data sets, they use different cell line and different approaches to perturb MND pathway. And they use different sequencing technology to identify so-called MND substrates. As you can see from this Venn diagram, the substrates, the MND substrates, they identify from these 3 independent labs. It's not totally overlap. But when I reanalyze those publicly available data sets and pulled out those MND substrates, I asked solely one question: whether there are any pathways that enrich from these so-called MND substrates. And amazing enough that I found a bunch of pro-inflammatory pathways that we all hate. It's enriched here. ALPS stimulated MAP kinase pathway, toll-like receptor signaling pathway, CXCL8, IL-1 signaling pathway. However, there are several caveats for those reanalysis that I did that I just showed you. First, those data sets, researchers use knockdown or knockout approach to perturb MND. So I don't really have time resolutions to tell you guys whether the effect that I saw, it's really the first hits of MND perturbation, or it's a secondary, or it's a tertiary response. Also, those data sets, the whole experiments was designed to look at MND substrates. So they don't really look at the whole transcriptome response to MND perturbations. But I think it's important that we fully understand what happened, what's the transcriptome response after we perturb MND pathway. And also that we already know MND pathway has a differential activity in different cell type or different tissue origin. So with all these caveats, I think it's critical to know the transcriptome response in different cell types or different cell lines upon MND suppressions. However, such knowledge is lacking. So the approach that I took is I used that SMOG1 small molecule or SMOG1I, and to inhibit MND pathways in 4 different cell lines from 4 different tissue origins, followed by transcriptome analysis. So the 4 cell types that I use are hepatoma cells, HeLa cells, HEC293 T cells, and myoblast. And the PCA plots that I'm showing you on the left here, I'm plotting PC1 over PC2. And as you can tell from these PCA plots, that basically all the data points or all the variants just cluster by cell type, not by the treatment effect. And in fact, up to PC4, that up to 80% of all the data variants can be explained by the cell type. Apparently, that's not the effect or the analysis that I want to look at, because I really want to look at the SMOG1I treatment effects across all cell type. To do so, I use this statistic framework. I use the SIG2 analysis with the formula. So right now that I can account for the cell line effects and only focus on the treatment effect. And with this statistic framework, I can have a PC1 account for 43% of the data variance, and that perfectly separate the DMSO treatment control and SMOG1I treatment at 24 hours time points. This is the analysis that I want to look at. I want to use this statistic framework to look at all the cell type and all the cell lines simultaneously and tell me what's the transcriptome response upon SMOG1I treatment. After I confirm the statistic framework that I like to use, I perform differential transcript expression, or DTE, for these 24-hour time points. And the volcano plots that I'm showing you here, every single dots represent a unique transcript. The red dots that I highlight in these volcano plots, they all fit into this highly stringent criteria with absolute log2 fold change bigger than 1 or super stringent FDR cutoff. And even with this stringent cutoff, I still get over 17,000 transcripts that is differentially expressed upon SMOG1I treatment at 24 hours. As you can tell, most of those differential expressed transcripts are regulated by SMUG1 treatment, even though some of them don't regulate it. And as you can imagine, that most of those upregulated transcripts, they are either aberrant transcripts with PTC, or they are normal transcripts, but they have long UTR or upstream open reading frame, so they still target by MND. And of course, with this 24-hour time point, it's likely that I also collect some signal that they are not direct targets of MND. And the volcano plots on the right, it's the same data, it's the same plots, but I just overlaid another color, the blue color that I'm showing you here, with the same stringent cutoff. So all these blue color transcripts, they actually are aberrant transcripts according to this database, MND transcript database that just published this year. And in this database, they use algorithm to identify all the transcripts to see whether those transcripts has open reading frame and look at the stop codon of that open reading frame. And if that stop codon is at least 50 nucleotides away from the last EJC, they will tag that transcript as aberrant transcripts because that fit into the canonical MND pathway to target that transcript. And as you can see, SMUG1I also increased a bunch of aberrant transcripts that it's highly elevated. And even though there are little tiny bits of transcripts that seems downregulated by SMUG1I, but overall I got over 5,000 transcripts that is aberrant transcripts that is upregulated by SMUG1I treatment. So with all this differential transcript expression analysis, the next step, what I did is I used all these differential expressed transcripts and perform pathway analysis again. The table that I'm showing you here, first it's 24 hours looking at all cell line, aggregate the data together. And here I selectively list several inflammation pathway. As you can see, all these culprits or all the molecules or all the pathway that we hate lights up here, toll-like receptor 3, MIT-ADA, NF-kappa-B, LPS-stimulated MAP signaling pathway, IL-6, IL-8, ERK MAP kinase pathway. They all have significant low p-value, enrichment p-value, and very high positive z-score. Z-score is a prediction score by the algorithm telling us whether this pathway is activated or not. So for all the pathway that I listed here, they have small p-value, meaning that they are enriched, and high z-score, meaning that the algorithm predict based on this transcriptomic data that the pathway is activated. And because this 24-hour time point signal is so significant, I decide to dial down at earlier time points. So again, I use the treatment for 4 hours and 8 hours compared to control group on HeLa cells. And in this table, I summarize the 8-hour time points and 4-hour time points together in this table and only pull out those pathway that I show you in the previous table that's still significantly enriched in this table. So for 8 hours, LPS or IL-6 PI3K pathway is still enriched, but only LPS and IL-6 signaling pathway with high z-score. At 4 hours, these two pathways still enriched, but they don't really get a very exciting z-score. So in summary, these time points transcriptome analysis telling us that MND suppression would activate inflammation pathways at early time points, as early as 8 hours, or even you can start to see some effects at 4 hours time point. And because this LPS stimulated MAP pathway is enriched throughout all 3 time points that I tested, so I would like to provide you more details about this pathway. On these pathway graphs here, all these red color or reddish molecules, they are the genes that they have transcripts that found in my transcriptomic data that SMUG1I treatment would upregulate their expression. And the green color means SMUG1I treatment downregulate their expression. And for those genes that I label with this stop sign, meaning that their transcripts, it's upregulated, and the annotation found that those transcripts are aberrant transcripts. So if I took off these aberrant transcripts from this enrichment analysis, the pathway is no longer enriched. That means both aberrant and normal transcripts contribute to the enrichment of this LPS MAP kinase pathway. This next plot, it's the same plot, same analysis, but I just overlay more colors on the top of it. Right now you see more orange color or orange line. These orange things, line or dots, they represent predictions from the algorithm. So the algorithm, if the algorithm predicts based on the transcriptomic data that this whole pathway or the line would be activated, then the algorithm would color this path as orange color. And if the algorithm predict that it would downregulate it, it would label it as blue. But as you can see, overall, that algorithm agree that this pathway would be activated or upregulated. And more interestingly, at the bottom of this path, it's CXCL8. That means the algorithm predict SMUG1I treatment would upregulate CXCL8 expression. And because that's a prediction, I really like to test it and confirm that's true, because CXCL8 is a well-known pro-inflammatory cytokine in CF lung, and that cause or contribute to the CF lung inflammation milieu. So in order to test that, I use this cell line, bronchial epithelium cell line 16 HBe 14O-. I expose the cell line to SMUG1I at 0.3 micromolar with or without Pseudomonas aeruginosa. And as you can see here, without LPS treatment, that SMUG1I already increased CXCL8 secretions. And even with more and more Pseudomonas LPS exposure, that co-exposure SMUG1I always increase more CXCL8 secretion. So this analysis tells us MND inhibitions lead to CXCL8 secretion by these bronchial epithelium cells, even in the presence of Pseudomonas aeruginosa LPS. And in fact, it's not just the analysis that I did telling me this story. There are other model organisms also telling us the same line. For example, SMUG1 heterozygous knockout mice has been found it will develop chronic inflammations and elevated serum cytokine levels as the CXCL8 level that I found. And also UPF1 knockout mice will develop this aberrant brain inflammation. And even in arabidosis, if you knock out SMUG7 or UPF1, it will get constitutive upregulation of pathogen response. So taking all together, that all this evidence telling us that we know non-MND or survey or degrade aberrant transcripts with PTC, or it also regulate normal transcripts with long UTR or upstream open reading frame. And when we try to hit MND to help CF patient with nonsense mutation, we also upregulate a bunch of aberrant transcripts and normal transcripts. And a bunch of those upregulated transcripts, they will turn out that flow into inflammatory response and exacerbates the CF inflammation milieu. So that tells us what we need to have. Maybe it's a transcript-specific MND inhibition, or we need to find a way to mitigate this SMUG1I treatment-induced inflammation response. With that, I would like to thank my crazy lab mates and also my boss, Suja, and Oscar and Kelly. They helping me to collect all this transcriptomic data. With that, I'd like to take any questions.
Hi, thank you. Great talk, but I'm just wondering about the details of the analysis. If I got it correctly, you said you see 17,000 differentially expressed genes. Is that right?
Transcripts, not gene.
Transcripts. How many genes would that be? So you did per transcript analysis.
How many genes? For
How
that?
many genes would they correspond to approximately?
If I remember correctly, that's about 3,000 genes. 3,000 to 5,000 in that
Now,
range.
if we look at your PCA plot, you see, well, we don't have to go there, but beautiful plot. It looks like it should. Everything clusters by cell type. That's the major variance in your data.
Right.
We know that when you do differential expression cell to cell, and it's really hard to do differential expression between different tissues.
Yes.
Because of so many differences, which typically amount to a few thousand genes, 5, 7, and at that point, it's already very difficult to normalize the samples. The effect per cell type of NMD inhibitor is very small. So how come that when you turn around your model, you see 70, that's the baseline. Cell to cell should be a few thousand genes. Now you look at NMD, and you see many thousands again, even though on the PCA plot, it's a relatively small effect. So your model, should there be an interaction maybe? Are you sure you should do just 2 main effects?
So that's the statistic model. It's not trying to find the common sense among all the cell type. It tried to find all the possible responsive transcripts.
It is trying to factor out the cell type.
Yes.
As a main effect. Maybe there is an interaction there. Maybe there is another term. Did you try to look at differential expression per cell type and then see if there is any commonality, any overlap? Are there the same genes going up and down per cell type?
Yeah, so that kind of analysis approach asking different questions. Those analysis approach trying to ask the common overlapping genes among all the cell type. But the analysis approach that I use, I'm not trying to look at the overlapping. I want to look at all the transcripts, no matter which cells that it made, as long as it responds to SMUG1 treatment well, the statistic model will capture that. And that's why I can capture so many
Or might
transcripts.
not. It could be a union of completely different set of genes
Yes.
expressed in different cell types.
Yes. So that's why it's important after I did this analysis, we always need to go down to use specific cell type that we interest, because whatever transcripts that the statistic model tell me, it just tell me, yes, this transcripts is response to SMUG1 treatment. But whatever transcript, it's a mixture of all 4 cell types that I test.
Thank you. That was really interesting. Thanks for that. I'm trying to make some broad links with what we heard from Anna just a couple of talks ago with the inflammation, and maybe you can comment on that. I can't remember if you said that you saw CFTR transcripts going up in your analysis at all, in your analysis. And then the other question I had was about how you're classifying aberrant from normal transcripts, because I mean, poison exons are a very well-known means of regulating gene expression, but they might be going into your aberrant if you're thinking about premature termination codons that are
Yes. So the second question, yes, it's going to the so-called aberrant transcript.
Okay.
Per that definitions. And the first question, whether CFTR transcript is upregulated, I faintly remember that I tried to look at that, and even though, yes, I got significant p-value, but the upregulation, it's not that dramatic as you think here. Yes.
Okay, and then I have one more. So is this probably like a natural pathogen response in some way that you would get this sort of increase or downregulation of nonsense mediated decay?
So you're thinking maybe it's part of the evolution thing.
Yeah. Are there any conditions, infections, or anything like that this happens naturally where you have? I'm thinking about like targets always, of course, for ways to identify novel targets. So if this is a natural response, it might happen that we can identify new targets for identifying ways to modulate nonsense-mediated decay.
That's an interesting thought, yes. I never thought about that. Yeah.
Yeah, I don't know if some pathogens might hijack the response and want to downregulate it so that it can produce more of it.
Yeah, for that sense that a lot of virus trying to do so, yeah. But virus mainly try to escape from the MND surveillance so that they can replicate. But I haven't came across any idea whether virus will try to hijack this system and so that downregulate the immune response. But I think that's a very intriguing hypothesis that virus.
The cell might have evolved to then have this inflammatory response
Yeah.
when that happened, right?
Yeah, virus just hijack this whole system to survive and also reduce the inflammation response.
Okay, one more.
Yeah, interesting talk. If I now bring the talk that happened before you, that talked about that the read-through efficiency becomes really good if you have an inflammatory response. And I think to treat CF mutations, what we are trying to say is a combination of NMD inhibitor and read-through. What would have happened if you had used any of the read-through compounds here and what you would have got? Because the previous speaker would be delighted to get to see this particular thing. This is setting up a stage for them to use the read-through.
Yeah, that's what I'm thinking after that talk. And I think that's, I hate to say this, but I think that's the beauty of CF, that somehow everything's go back to about inflammations, and we all hate inflammation. But from today's sessions, we already see several cases that maybe somehow this inflammation is actually helping us or helping the CF patient to go through this process.
All right, if there are no other questions, let's thank Zhang Yu and all the speakers for a great session.